H2S Analyzer, an AAI tutorial. In this video, we will cover the OMA H2S Analyzer, including the equipment that makes up the system and how the system operates. Overview. The OMA H2S Analyzer measures the real-time H2S concentration in a liquid or gas process by monitoring the UV-Vis absorbance of a continuously drawn sample. Analyzer and flow cell/sample conditioning system connections. Let's take a closer look at the OMA H2S analytical system. This is an OMA process analyzer with a standard wall-mounted enclosure. Inside the analyzer enclosure, there is an HMI unit, a Nova 2 UV-Vis spectrophotometer with a light source, and an I/O board, which is behind the HMI. Outside the analyzer enclosure, a flow cell is connected to the spectrophotometer using fiber optic cables. The sample gas goes through the flow cell for analysis. The light signal travels via fiber optic cable to the entry point of the flow cell, where a collimator narrows the light beam. The signal travels directly across the flow cell path length, interacting with the continuously drawn process sample fluid. The flow cell can be integrated into a customized sample conditioning system. Analytical principle. To measure the H2S concentration, the OMA detects the distinctive absorbance curve of pure H2S and mathematically isolates this structure from the total sample absorbance. In accordance with the Beer-Lambert law, the absorbance of a chemical in a mixture within a fixed optical path length is directly proportional to the concentration of that chemical. The OMA correlates the height of this curve directly to real-time hydrogen sulfide concentration. Different OMA configurations include the OMA 300 wall-mounted analyzer, the OMA 206 portable analyzer, and the OMA 406 rack-mount analyzer. The OMA 300 is offered in two explosion-proof formats, EXP and EXD. Sampling. Applied analytics systems are built for direct analysis without cooling or drying the sample. This allows us to build far simpler, more elegant sample conditioning systems that retain high sample integrity and optimal response time. The flow cell is integrated into the sample conditioning system. The length of the flow cell ranges from 2 mm to 600 mm. The path length of the flow cell is optimized in accordance with the Beer-Lambert law. For example, longer lengths are required for low ppm H2S concentrations, whereas short lengths are required for high percentage H2S concentrations. Safety. The key difference between an applied analytics H2S analyzer and other H2S analyzers is the use of fiber optic cables and external flow cells. We bring the light to the sample instead of bringing the sample to the light. The toxic sample fluid is only required to circulate through the dedicated flow cell and never enters the analyzer enclosure. Remote support. We offer remote support to your applied analytics analyzer. The OMA analyzer can be utilized in the following applications: natural gas, biogas and landfill gas, acid gas or feed-forward gas, sour gas, tail gas, hydrogen recycle gas, H2S scavengers, midstream hydrocarbons, wastewater treatment, and specialty and industrial gases. For more information, give us a call or visit our webpage.